understanding how structure impacts performance. So my background really is lipid biophysics and self-assembly, but this is extended to lots of different types of sort of drug carriers and drug nanocarriers over the years, so also including things like polymer systems. But today, the work that I'm going to present to you is really purely focused around lipid-based nanoparticle systems and how we might think about moving towards being able to couple some of the structures that we can understand and how that might link to performance. Just to give you a quick outline of the talk. So I'm going to briefly sort of give you a little bit of a background about nanoparticles, or what I mean when I'm talking about lipid-based nanoparticles, and sort of some of the sort of basic theory behind some of the self-assembly, some of the concepts that I find interesting. And then going to go into a little bit of a sort of case study, talking about actually studying bulk lipid formulations. And what I mean by bulk in my case is really looking at just the lipids themselves, not in nanoparticle form, and how they behave under different conditions. And then moving on, we're talking about some of the population analysis studies that I've been looking at more recently. So one of the things I find fascinating, if you look at the sort of range of lipid nanomedicines that have actually made it into the clinic, is actually, although there aren't a huge number of them, they span a huge range of diversity. So for example, there are bulk formulations of the clinic, which come from the Swedish company Camurus, looking at a slow release of drugs for treating opioid addiction. And the way this formulation works is really you have a self-assembly of the bulk lipid phase when it's injected under the skin, and that this then leads to a slow release of the active ingredients. And if you look at the sort of kinds of publications that come from Camurus as a company, there's quite a diversity of different lipids that they work with, but also the kinds of nanostructures that they're looking at. So they report a lot on, for example, inverted hexagonal phases or FD3M phases, so packings of micelles, basically. If you then look at the sort of lipid nanoparticle-based formulations or nanoparticles made from lipids formulations, so the first formulations that really have made it into clinical practice are these liposomes. And these sort of pegylated liposome formulations are used for chemotherapy. So, for example, doxorubicin, delivery of chemotherapy drugs, but obviously the sort of most popular aspect of nanoparticles made from lipids at the moment is referred to the sort of lipid nanoparticles in the COVID vaccines. And really here, the first thing to come onto the market was actually Onpattro, and I see now already that I have an mRNA mimic in my Onpattro image, which is not correct. It's an siRNA lipid nanoparticle formulation for treatment of amyloidosis. And we do actually have mRNA in the COVID-19 vaccine. So the spike vaccine, the Comirnaty thing. And here I've done a sort of illustration of the kind of phase separated structures that you see in some of the cryo-EM. So one of the sort of key factors in all of these medicines is that lipids are responsible for the packaging and delivery of the active ingredients. And if you look at the sort of different components that are worked with, generally there tends to be some sort of ionizable or cationic lipid. So something that can either respond to pH changes to become charged or is charged from the start in order to facilitate delivery of payloads. There are some sort of lipids that help to form some of the structure or protect some of the structure of the nanoparticle itself, often referred to structural or helper lipids, and this could include things like phosphocholine, cholesterol, those kinds of lipids. Basically, the majority of the nanoparticles, certainly that made it into clinic, have required some level of stabilization. So this could either be through the form of pegylated lipids, which is what the lipid nanoparticles in the COVID vaccines have, or some sort of stabilizing polymers, and then having the payload itself. So for example, here are some different types of RNA that I've illustrated. And I think one of the really things that fascinates me about this is that depending on which lipids you pick and how you self-assemble your system, you actually end up with really quite different structures and types of nanoparticle at the end of your process. So you could, for example, have a very amorphous core with a monolayer surface. You could have a much more structured core, for example, with a bilayer surface or a monolayer surface, or you could have something more similar to these sort of phase-separated structures. And actually, in many of the cases, there's not always a huge difference in lipid composition between these types of structures that can be formed. So my background is lipid biophysics and lipid self-assembly. So one of the things that really fascinates me about all of these type of structures is that they actually span a really large range of membrane curvature and interfacial curvature. So when we tend to think of bilayers or membranes, we often tend to think really much more about these sort of flat surfaces, what we would call a lamellar bilayer. But actually, when we mix lipids together, we can form a huge range of different complex structures. So we have, for example, the bicontinuous cubic phase structures. These still contain a bilayer, but what happens is the bilayer is really draped over an infinite periodic minimal surface. So what we have is you can see here in the sort of red and blue with this nice image that I've borrowed from this PNAS paper, you can see two interconnected water channels that we have in all of these different kinds of structures. There's also the hexagonal phase. In this particular case, what I've shown is inverted hexagonal phase. We have almost like these cylindrical structures of lipids with water channels coming through the center. We have packings of micelles in various different structures in different space groups, and then all the way up to sort of inverse micellar solutions or even amorphous mixtures where we have very little structure at all. And if you look at actually where the medicines that have made it into the clinic sit on this spectrum, they actually really span a huge range of self-assembled systems. So this goes all the way from working with liposomes, which form flat, hopefully single bilayers, all the way up to, and this is a little bit of a guess for the Kimura's formulation, but it's based on what they've published, up to sort of packing of much more micellar structures, all the way up to the sort of Onpattro clinical formulation or the COVID vaccines, which if you look through the literature, you can actually find quite a diverse range of different structures or different discussions about the types of structures and arrangement of lipids that we have in these different particles. And in many cases, this could be due to slight differences between formulation, for example, changing the ionizable lipids or changing the payload. And in other cases, it's maybe less clear what the reasons for the differences in structures are. But there are, if you sort of even just go and sort of trawl through cryo-EM images of the different particles, there are many different structures that can be observed in actually quite a narrow range of composition. So one of the challenges but exciting things with self-assembled systems, and that I find fascinating, is that there are many different factors that influence structure. So this image came from a review that we wrote back in 2019. And in this particular system, we were actually focused on structured lipid nanoparticles called cubosomes. And basically, in this particular case, what we have is on the internal portion of the nanoparticle, we have a sort of interconnected lipid bilayer in a bicontinuous cubic phase structure, and then an interface that is enriched in some sort of stabilizer to stabilize the particle. But the sort of real essence of this image in the first place was actually to try and illustrate which factors influence structure if we were to use these kinds of particles, or, for example, in other less structured particles for pharmaceutical delivery. And what we sort of really wanted to try and highlight here was that almost anything you do or put in a self-assembled system will change its structure. Whether that's important or not is almost the next discussion, I guess, but it will cause some sort of changes in most cases, I would say. And so what we want to illustrate here was, for example, work that tuning the lipid composition, so changing the amount of charge or the bulkiness of the tails, so how many tails that we have in our system, the kinds of things that we use to stabilize the particle surface, so the kind of stabilizer like a pegylated lipid or a polymer-based. If we want to start loading these kinds of particles for pharmaceutical application, how we do that and where we put them. So, for example, are we interested in loading into the water content of the particle? Are we interested in loading into the sort of bilayer lipid content of the particle? And then actually adding in sort of biologically derived compounds such as DNA, RNA, coupling to the lipid membrane, or even inserting proteins into the system. And in addition to that, where we have to really think about what the different components that we have in our system are, there's also quite an impact to our systems on formulation conditions. So these are two studies, they're quite fundamental studies, but what we were looking at in the first case really was how a specific enzyme was able to chop up different portions of specific lipids and how this changed the structure. And what kind of came out of this study almost as a side product was that we realized that if we made particles of exactly the same lipid composition, either by mixing those lipids together and formulating them, this was via tipsonication, or by mixing a different set of lipids together and using the enzyme to edit the lipid composition of the particles, that actually we ended up with particles of the same composition, but very different structures. And that was a sort of unexpected, I would say, or not, maybe not surprising, but it wasn't something that we were looking to find as part of this study. We've also done some studies looking at formulation of different vesicles and whether they are sort of unilamellar, bilamellar, so whether they have several bilayers, what I mean in this, and the sort of formulation process, and seen that, for example, even the salts that we have in there in these processes make a huge difference to the kinds of structures that we're able to form. So one of the challenges that we have working with lipid nanoparticles, so I've sort of moved much closer to the clinic now in terms of thinking about different types of nanoparticles, is actually that they themselves have quite what I described as a complex journey from benchtop to clinic. So it's quite widely well known, but to formulate these particles, we tend to have all of our lipids in a solvent, predominantly ethanol is used. We have our RNA payload in buffer, generally at a low pH. There's some sort of rapid mixing that takes place. So, for example, via a T-junction, via a herringbone microfluidic mixer. Some people have reported even pipetting steps to be able to do this in high throughput. And then you collect what comes off at the end of your rapid mixing process. And then there's a dialysis step. And this dialysis step is predominantly to remove the excess solvent that we have in there, because we've mixed these generally in different ratios. So there's a certain portion of solvent, often around sort of 20–25% of solvent in there, and to bring the pH up to a much more neutral pH. So some groups do this, for example, by putting this, by doing an overnight dialysis, either at room temperature at 4 degrees, and other people do a sort of much faster concentration dilution step. But either way, there's a sort of maturation of the particle and a buffer exchange. And then once you've got your final particle, the debate is then how do you store it? So you can either keep it at 4 degrees and they'll last a certain amount of time, but specifically you're working with mRNA, there'll be degradation that takes place. And so there tends to be often an additional processing step where we see these particles concentrated and then often put into some sort of storage buffer and then frozen and then thawed again. And this actually means that, considering we're working with quite complex self-assembled systems, they've gone through a whole range of different processing by the time they're actually going to be used at the end. So one of the things that I'm interested in is trying to understand what happens along all of these different stages and sort of what is important for performance. And so the way that I'm kind of a lot of the work that I'm doing at the moment approaches these questions is really to try and combine different analysis approaches. So we have the bulk analysis approach, and what I mean when I talk about bulk is really looking at the lipids themselves in isolation, so not as nanoparticles, but how do they behave in different conditions by themselves. We then look at the sort of overall average behavior of our of our formulation. So if we take our lipid mixtures and we formulate them into particles, then on average, what is our formulation like? And I'll go into a little bit more detail by what I mean in that case. And then we're now also trying to narrow it down to the single particle level. So we understand on average what our formulations are like, but down at the single particle level, do our formulations look different? Are they performing differently? And what can we learn from really understanding the single particle level? So the next portion of my talk, I'm going to talk a little bit more about some of the sort of lipid bulk work and understanding structure in lipid systems that we've done there. So this is a sort of side note to the bulk work, but I would say that generally this is not something I'm going to go into detail today talking about, but there's a sort of fairly standard sort of quality assessment criteria that we use for pretty much all nanoparticle systems that we're working with. And what we're really thinking about there is like, what are the sizes of our particles? Are they reproducible? What's the polydispersity? So what's the size distribution that we have in our particle formulations? What kinds of concentrations of particles are we working with? What is the surface charge, so zeta potential of our different systems that we're working with? And then also, how well have we encapsulated the payloads that we're working with? So in this case, by encapsulation efficiency, I mean how much was left in solution and how much was encapsulated in the nanoparticles that we were looking at. And so they're not sort of parameters that I'm going to dwell on in this talk unless they're relevant to the specific topics. But as a general sort of rule, we would always kind of do a quality criteria assessment on all of our particles that we're working with, not necessarily always to include or exclude them, but to just be aware of what the quality of our particles is and how reproducible that is. And so a lot of the work that I do is based on using small angle scattering to understand the structure in different systems. So in Sweden, we have, I'm sure everyone, or many people are aware of this, but we have max for.
Na przykład mieszaniny tych czterech i dopiero wtedy można uznać, że nawet jak one wszystkie wyjdą pi razy drzwi w tym samym miejscu, to albo mamy tą...
So we have max 4 down in Lund, which is a great facility for performing X-ray, and also the ESS, which is currently still under construction down in Lund. And I also have worked quite closely with Diamond and in the past ESRF and also ICIS neutron and muon source to do X-ray and neutron scattering work. So I think all of the work that I'm presenting today was performed at Diamond at ICIS, but that's just potluck, maybe a little bit at ESRF actually, now that I think about it. But with the small angle scattering, I'm not going to really talk about the theory of small angle scattering today, but just in case anyone isn't as familiar with it, basically the idea is really that we have our beam of X-rays or neutrons. We go through various different sort of focusing and filtering or processing of the beam so that we have the right sort of energies and collimations by the time we get to the sample. And then once the beam hits our sample, it can effectively sort of be transmitted, absorbed or scattered. And we're predominantly interested in the scattering. And this is collected on a detector. And learning about detectors for small angle scattering is a whole fascinating science in itself. But basically, if we're lucky, we get a signal on the detectors, and what we tend to then do is we do a radial integration of our sample and then look at the data as a function of the reciprocal space. So one of the reasons that I enjoy working with X-rays and neutrons, and actually also cryo EM, is really that we can exploit these different techniques to get study our systems in slightly different ways. So this is a really nice picture of an old medieval sword, and then it's been imaged both with X-rays and neutrons. And what you can see here is that although we're looking at exactly the same sword, you can actually see really huge differences in the kinds of contrasts that we're getting from the different systems based on how both X-rays and neutrons interact with different properties of the system, so with the different compositions of the system, basically, so with the atoms. And where I'm interested in doing this specifically for my work is looking at studying structure and function relationships, specifically related to lipid nanoparticle systems. So some of the studies where we've used these kind of techniques include to look at enzyme dysregulation. So in many different diseases, phospholipases are dysregulated, and phospholipases are a specific type of enzyme that can chop lipids up in a specific manner into different pieces effectively. And what happens when you change those lipid compositions is actually you change the phase behavior of the system. So in these two systems we were looking at, we found that actually in one case, the phospholipase was able to completely destroy liposome systems that we were looking at. And this was actually due because we ended up with a phase transition. And the sort of system just became, if we had just had a bulk system, then I think we would have just seen this nice phase transition. That's what we saw with the bulk. But as we were going from one type of nanoparticle to another, actually the whole thing collapsed and we weren't able to detect our liposomes anymore. And another study where we've looked at this similar idea but completely different lipid formulations was we actually found that in this particular system, the reason that we were able to detect lipase activity was actually that the lipase was causing a phase transition where we went from a fluid to a gel bilayer, and then we had a leakage across the bilayer during this system, during this transition. I've also done a lot of sort of fundamental work about how lipid composition drives structure, which I'm not going to go into more today, but although some of those studies can often be quite abstract, they can then lead to much more sort of complex studies with many more complex lipids. And something I've been focusing on a lot more in the last couple of years is using these kinds of understandings to look at ways to optimize delivery. So can we understand why certain delivery nanoparticles behave in a way they do? And are there ways that we could look at understanding how to improve them? So, for example, in this final study, what we saw was that when we had cholesterol in our system, we had a much higher propensity for phase separation. And then actually the release properties were much, much faster in that particular system. And it was a triggered peptide release. So one of the things that really fascinates me related to nanoparticle design is this ability or inability sometimes to tune the lipid structures that we're working with. So I started working with lipid self-assembly on some much more fundamental studies. And what we were really looking at in those fundamental studies was looking at tuning the sizes of specific lipid structures. So we were looking at bicontinuous cubic lipid structures, and what you can see here is a sort of image of the unit cell. And what we wanted to do was to be able to swell the sizes of the structures from a small structure to a larger structure. And in this case, predominantly what we were really changing was the sizes of the water channels, although it's possible that there were also some small changes in the membrane thicknesses that we were looking at as well. And so what we saw in this study was really that as we increased the amount of charged lipid in our systems, providing that we had cholesterol in our systems to stabilize the system, we were able to swell the systems quite dramatically. And actually the ability to swell them this much sort of went beyond the limits of what we had thought would be theoretically possible, actually. And what we found was that at some point we reached the tipping point, and then we had a phase transition, and then we ended up with lamellar structures instead. And this was really a very fundamental soft matter study, swelling and shrinking bicontinuous cubic phases. And we saw that they were very sensitive both primarily to the electrostatics in the system, so the amount of charged lipid that we had in the system, but also to temperature and pressure, and that they didn't necessarily behave in exactly the way that we thought they did. But so one of the things I found really interesting about this system was then whether this would translate to nanoparticles, because it was at a time where it was becoming much more popular to turn these kinds of complex structured systems into nanoparticles. And because they have quite unique properties where you have inside the nanoparticle, you have both aqueous water channels, so you have solvent, quite a lot of solvent, and also a continuous lipid membrane. It means that you can kind of pack drug molecules or similar kinds of molecules in a slightly different way than you could compared to, for example, a vesicle. And so I set about trying to repeat the study, but translate it over to nanoparticle format and to see if actually the engineering rules and the way that we could tune the structures in a sort of bulk paste format also translated to nanoparticles. So we didn't necessarily expect to get exactly the same results. We have to formulate the nanoparticles in a particular way, and we also have to stabilize them. And so we were expecting that that would kind of shift the phase boundaries a little bit. But generally what we found from this study was that the electrostatic tuning rules that applied to bulk lipid structures also translated into nanoparticle structures, with the caveat that you had to make some allowances for the differences between them. And the result really of being able to do this was that what it changed by changing these water channel sizes was that you changed access to the internal structure. So this meant that certain molecules were then able to flow, and we validated this with some FRET, but certain proteins were able to access the internal core compared to not, depending on how we tuned the structure. And this actually turned out to be useful in many other areas of research, specifically actually for protein crystallization. So lipid cubic phases, and particularly bicontinuous cubic phases, have been used to promote protein crystallization, specifically with various different membrane proteins. But one of the challenges of being able to crystallize proteins with very large extracellular domains was purely that they didn't fit. So the water channels were simply too small for protein crystallization to take place. But by using the same actually engineering rules that we'd used in the bulk studies, what they did was that they managed to swell the bicontinuous cubic phase. And this gave enough space for these large extracellular domains to be able to diffuse through the lipid system and then crystallize. And actually, this sort of principle of looking at how bulk lipid phases behave and relating this to nanoparticle behavior is also being used now for lipid nanoparticle research in itself. So there was a nice paper that came out in 2023 with AstraZeneca were one of the co-authors from Joachim Redler's group as well. And what they did in this particular case was they were looking at the different bulk phases of the lipids from lipid nanoparticles. So they took the lipid compositions of the core and looked at how those varied as a function of pH, ionizable lipid, and payload to look at how the structures differed under these conditions to try and provide a mechanism of action of how the different lipid behaviors were impacting endosomal escape and release of payloads. So I think this is a really nice example of how these much more sort of fundamental bulk studies can really translate all the way through to clinically relevant lipid nanoparticles. So one of the challenges when we're working with nanoparticles and bulk lipid mixtures is always analysis of our data. So I've drawn this really nice sort of little yellow blob to sort of illustrate lipid bulk phases. But if we're working with highly structured phases and we do X-ray scattering, then often we have very defined structures with well-defined peaks, well-defined intensity ratios between the peaks, and also spacings between the peaks. And this enables us to do a very accurate phase assignment. In reality, when we break these systems down into nanoparticle systems, firstly, we have many fewer repeat units. We have generally or often very small nanoparticles. The core of the nanoparticles is very often not structured. It could be very unstructured. It could be very amorphous. And also we have this sort of distribution often of structures across our nanoparticle formulations. And so in reality, what we get out when we do these kinds of structural studies on nanoparticle systems is, as I've had described to me, squiggly lines. And so one of the challenges that we face in the field sometimes is really trying to interpret the data that we get out. All of the information is in there, but how do we extract that information? And how do we relate that back to the sort of physical properties of the nanoparticles themselves? So one of the ways that we've tried to do this is actually by combining, for example, neutron and X-ray studies to really look at the different techniques and see what we can learn from the two of them. So this was a study that we performed with a structured lipid particle system. And what we were able to do was to effectively try and mimic the X-ray study, where we knew we could tune the internal sizes of the lipid particles and see that if we fitted with specific models in the neutron data, so this broad peak model, were we able to reproduce the kind of behaviors that we knew the systems were performing from the X-ray data. And so the reason we were interested in doing this, I guess, was here we have these broad peaks and a little one peak here. And this is not really enough for us to be able to say what kinds of phases that we're dealing with. So what we wanted to see was just really, could we reproduce the behavior, so at least in terms of sizes, that we could make a sort of accurate prediction that these were the kinds of phases that we were looking at. And they reproduced really nicely between the different techniques. And so one of the things that we're kind of thinking about a lot now, particularly for a lot of the neutron data that we have, is how do we approach fitting of the data? So if we take 4 different LNPs and we measure them, in this particular case, the LNPs are quite large, so we really don't see that much of the Guinea region here. But we have kind of 3 different ways that we can approach actually handling this kind of data. So one is a sort of shape independent models. And if we do that kind of fitting, what we're kind of doing is really trying to assume as little as possible about our system, and then trying to understand what we can extract from those fitting. The other option is to use shape-dependent fitting approaches. So this is where, for example, we assume that we're working with a sphere, or working with ellipse, or working with a cylinder, and see how well those specific models fit to our systems. And then the third one, which is maybe not quite so established at the moment, is the idea of being able to use simulation guided or model guided approaches to being able to fit our systems. So what here are some sort of illustrations, but we have some sort of basic scattering functions that we can use that assume very little about our structures, although they do still make some assumptions. We have the shape dependent, which are a sort of much more popular way of fitting lipid nanoparticles, where you assume often a spherical particle, and then you add different levels of spheres on them to be able to try and fit the data. Or we can try and use where we try and create starting structures and then see if our structures that we construct match the data that we're working with. And I think even actually one thing that I found particularly interesting is that even by using very basic models, so some of the shape independent models where we really assume very little about our structures, we can see quite a lot of differences still that come out. So in this particular paper, we were mostly interested in the internal structure, actually. So this particular region, so most of the plots in the main publication kind of focus on that. But if you dig a little deeper, we actually saw quite a lot of differences, for example, in the slopes here. And when you plot out the Poirot exponent, what we can actually see is that as we were formulating our particles with different lipid compositions, we were also not only changing the composition of the particle, but also the surface structures of the particles that we were working with. And then the sort of additional challenge that we have in this case is that what we see when we're doing neutron scattering is generally an average of the samples, or is an average of the samples that are really being measured by the beam. So we're really sort of measuring everything that's in there. And this is great because the average sample is what we use in the end, but that doesn't enable us to kind of break down to an even smaller detail what the differences are in some formulations. So one of the things we've found, for example, when we've been looking at more structured systems with cargo in them, is that when we add cargo, we see shifts in some of the peaks and not all of the peaks. And this, as a sort of scatterer, really implies that some structures are changing and other structures are not changing. So we had quite a few different theories about this and what this might be, but generally it's sort of evidence that we do have subpopulations in the structures that we're looking at. So different types of populations in the nanoparticle formulations that we're looking at. And so for the last sort of section of my talk, I'm going to talk a little bit more about some of the population analysis that we're trying to do to really understand the different types of nanoparticles that we have within, for example, a single formulation. So to continue from the slide that I showed previously, what we actually found in this particular case is, and when we started to look at the number of proteins that we had in each particle in this case, we did actually see differences between, firstly between formulations, but also that there were some particles that simply had no protein in at all. And so what happens when you sort of start thinking about this in the context of mRNA, for example, loaded lipid nanoparticles? So I said at the start in a sort of quite simple fashion that we do always think about payload encapsulation. And when we're talking about payload encapsulation, we predominantly work, for example, with like a fluorescence assay, like the ribogreen assay. And what we're doing in that particular instance is we're looking at encapsulated RNA as a function of the total amount of mRNA in the system, and then we're plotting a percentage of it. And so what that really means when we start to think about it is that what we're really looking at are the different ratios of free mRNA that we have in our solution versus the mRNA that we have encapsulated in particles. But really the kind of information that we're not getting from this kind of assay is much more related to what kind of distributions did we have. So you can see here that I've drawn some empty particles, I've drawn some particles with a lot of mRNA in, and I've drawn some particles with very little mRNA in. And this is just an illustration, but we really don't know if this is the case or not when we're just looking at an encapsulation efficiency in our samples. So this is actually where we teamed up with a collaborator. So he's Erdinc Seskin is a PI at Karolinska Institutet, and he had developed a technique for analyzing single particle fluorescence. And basically, this is a sort of extension on the FCS technique in the sense that what you are able to do in this technique that you're not able to do in FCS is that for any individual particle, you're getting both channels of fluorescence out. And this can be performed on a confocal microscope in a 96 well plate format. So we kind of contributed some nanoparticles to this study to really see if we could use it for lipid nanoparticle analysis. And as part of that, what we had to do was we had to work with both labeled LNPs, so we had a lipid dye in our LNPs, and we also had to optimize mRNA labeling in our LNPs. And we had to be confident that we had a full labeling of all the particles that we were working with. Then what we did is they measured a bunch of different samples for us. So we were specifically interested at that time at looking at structured lipid nanoparticles with mRNA in them. And what you can see is that they then measure in two different fluorescence channels for a single event. You're able to see what the fluorescence of the two different channels are. And then if you look at the kind of data that we get out from that, you can start to distinguish what kinds of particles they're potentially picking up. So for example, if we're picking up both channels at the same time, it's likely that we have an LNP with mRNA in. If we're just picking up the LNP signal, but no mRNA signal in that particular event, then we've got an empty LNP. And if we're just picking up an RNA signal, but with no lipid signal, then we've got an RNA aggregate in solution. And what this enabled them to do was actually to be able to look and detect different populations in the formulations that we were looking at. And actually, they've since done some further development work. So now that they can be a lot more confident as well about what the actual numbers of different molecules are in the different cases. And so what we learned from doing this study was that in these particular formulations that we were working with at the time, there was a huge variation in mRNA distribution in single LNPs within a single formulation. So in this particular case, we found that actually a huge number of our LNPs were empty, and we did look at different cationic lipid ratio to mRNA ratio. And we also saw that if you looked within any distribution, not only were there more or less empty LNPs, but also the number of mRNA or the amount of mRNA across the different particles followed a distribution as well. And to complement that study, we also added in an additional membrane dye that we were looking at in our lipid nanoparticles, and saw that if we made our lipid nanoparticles with different amounts of cholesterol in them, firstly, we were able to detect different signals from the fluorescence in this particular case. But also, if you look for any particular batch of lipid nanoparticles, you can see where every single one of these dots is a measurement. There's quite a broad range of membrane biophysical sort of properties that we're picking up in this particular case. So we moved on from this to work with a technique that was developed in the Stevens group. So it basically is called single particle automated Raman trapping analysis. And the principle of the technique is really that there is an optical trap, and within that optical trap, in an automated fashion, you can trap single nanoparticles, and in size they vary from about 50 up to a couple of 100 nanometers in diameter. And then once you've trapped your single nanoparticle in your optical trap, then you're able to take a Raman spectra of that particle. And then what happens is that in an automated fashion, you can then drop the particle and wait for the next particle and measure that one. So this means that over a period of sort of, well, it depends how long you measure for, but over a fairly short time period, you can build up a picture of what single nanoparticles look like by sampling sort of hundreds to thousands of particles within your solution. And so we use this basically to look at different lipid compositions of particles in solution, but also to do some dynamic reaction monitoring, where we're looking at an enzyme chopping up various components, lipid components within the system. And what we can do with this technique, which is quite nice, it gives us the option to combine average and single particle data related to lipid composition. So the spectra that you can see here are average spectra taken over the whole set of measurements. And in this particular case, what we were doing was looking at the difference between a phosphocholine and a phosphatidic acid. And what you could see is that there was quite a unique signature as we changed the ratio between these two lipids that we were able to detect. And then if you went and did basically the same type of analysis, but for single nanoparticles, what you can see is that in the formulations we were working with, we saw quite a wide distribution of lipid composition between different particles in our formulations that we were producing. And so what we're doing with this moving forward is really applying this technique to be able to look at, for example, more clinically relevant lipid nanoparticles with mRNA in. And so one of the things we've been interested in as well, can we look at, for example, surface changes of LNPs? So this technique in itself is not distinguishing between the internal and the surface Raman spectra, because it's just within a single confocal volume. But we're doing, for example, in this case, what we were doing was we're adding FBS to LNPs and seeing if we could detect the protein on the surface. And in this particular case, we also do a background subtraction. So we're not taking into account any FBS that might be left in solution. So firstly, we tested a few different LNPs, basically to see if we were actually absorbing any protein onto the surface. And in some cases, we were absorbing more than others. And also the size distribution wasn't completely changed by the absorption of protein onto the surface. And then what we set about doing was using this technique to try and quantify the surface protein. So if what you see here on the left are actually the average spectra that we've taken of different formulations. And then this work is done by a really great PhD student in the group, Lee Ma. And what he's looked at here is he's looked at the LNP signals. So are the lipid compositions or the LNP signals that we're picking up over time after incubation changing? In these conditions that he was looking at, they weren't changing significantly, but he's also then able to differentiate what the FBS signals are. So if we start looking for the pristine LNPs, which are LNPs with no FBS, you can see that there is some level of background that we do picking up, or some level of sort of variation in the measurements, but that overall, actually, we can start to detect quite strong FBS signals on individual lipid nanoparticles in solution in this case. And actually, if we enhance responses signaled by labeling, we can then shift even more and really change our limits of detection by looking at the individual particles. So now I've kind of outlined the majority of what I was going to talk about, and I've kind of presented them really as two quite abstract cases where we're really looking at the sort of bulk average detail related to structure, and then we're going much more down to the single particle level. And I think what would be really exciting was be able to sort of marry the two and really relate these single particle to the structures that we're seeing and being able to decouple. This is really a challenge with soft matter systems. We have a sort of limited number of models available to us for structural analysis. And so if you want to do anything more complex, you really have to start building your own. So there's an exciting project that's being led from some collaborators in the UK. So it's called the Shapes Bio project, and it's a sort of novel workflow to enable new opportunities in soft matter for data analysis. And it's called the Shapes Bio project. I've been collaborating with them on this. And the idea here is really that you can start thinking about generating different kinds of structures to sort of be able to model what your systems might look like in theory, and then see how that relates to the scattering patterns. And so here's an example for example, SDS. This is a quite basic example, but what the sort of workflows that they've developed enable you to do in Python is really to develop the individual molecules and then to tessellate them on surfaces. And the idea is that this will be expanded to more complex structures, so that further down the line, you could imagine a situation where you were able to generate starting structures and try and understand how the scattering is different in different cases. And this will really enable us to start linking up these sort of bulk average structures to the much more atomistic details. I want to say a huge thank you to all the people that have contributed to the different work. I've shown the work specifically from two PhD students, Lee Ma, so he's currently a second year PhD student, and Catherine Saunders, who's actually graduated now, as well as many of the collaborators that I've worked with, both on Beam Times and for different collaborators, Molly Stevens, and also all the Saxon Sands Beam Time teams that have come and worked with me, both from Karolinska, Imperial, and other collaborations, as well as my collaborators at the infrastructures and also the funders. And as a small little plug, I'm currently recruiting for PhD and postdoc positions. So if you're in a position that you're looking for something, some new avenues to explore, then please feel free to get in touch with me. Thank you.